Well, thank you, Mark, for introducing me. That was a pretty thorough overview and also kind of leads into my talk very easily. I'm talking about the dark side of the carbon cycle, which I refer to as respiration because it occurs not only when it's dark, but throughout the day, as opposed to photosynthesis, which is the light-driven reaction. And when we think about this globally, I'm going to stand so I can point. Photosynthesis on an annual basis assimilates about 120 gigatons of carbon into the terrestrial environment. And approximately half of that, or 60 gigatons, leaves through plant respiration from leaves, stems, and roots. And again, an equivalent amount leaves through soil respiration. And this is on the same order of magnitude as this, which occurs in the ocean systems. Is this advancing? Yes, okay. So to put this into perspective and really think about how big a flux respiration is, as Mark mentioned, it's less studied and less thought of as being environmentally sensitive as photosynthesis, but it's a gigantic flux. And it's nearly 6.5 times the fossil fuel emission, land use change, and cement emissions globally for every year. So this is a gigantic flux, so even small environmental variability in plant respiration would overshadow smaller changes in these fluxes, which we pay really close attention to. And this is especially important because of the extreme rapid change we're experiencing through climate change. And this is happening not only in terms of temperature. Here you can see this is a pretty recent graphic from Climate Central showing that 2016, which has been a very strange year for many reasons, one of which is it is giving us and representing the highest recorded temperature since we've had the instrumental record in temperature. So each month in 2016 has been the hottest on record. And then also, when thinking about carbon dioxide levels, we've recently surpassed 400 ppm. So this is kind of a mental threshold, if not like a really physical one, in terms of the fact that we are in a very high CO2 environment compared to even the recent past. Sorry. So despite this enormous flux, so we're talking about 60 gigatons of carbon entering the, or released from the terrestrial environment through plants every year, these controls are not well understood. And this variability in respiration can determine whether the land surface becomes a CO2 source, emitting more carbon dioxide than it's taking up, or a CO2 sink, where more is being assimilated. And respiration, as Mark mentioned, is really sensitive to the environment, potentially more so in some situations than photosynthesis. But unfortunately, unlike photosynthesis that has an equivalent, or it has a mechanistic model that's been well described and also scales from the leaf level to Earth system models, the Farquhar photosynthesis equation, respiration kind of lacks this mechanistic model because there's so many different environmental controls that are kind of complicating its ability to be simplified. And so generally, this poses a challenge to describe respiration at both global and ecosystem regional carbon models. So just to give a backdrop for those who haven't thought about the Krebs cycle for a while, mitochondrial respiration, which is generally what I'm talking about, is again this kind of dark process. It occurs in all cells, in all tissues, at all times. So in the light, in the dark, in roots, in leaves, and stems. And generally, it's a huge energy-producing pathway where sugars are modified in the cytosol through glycolysis and then enter the mitochondria in a different form, go through the Krebs cycle, and then at this point, this carbon dioxide is released. And this carbon dioxide globally, again, accounts for 60 gigatons of carbon annually. So even though we're thinking about these very small cellular processes, the composite flux is gigantic when we think about the global carbon cycle. So what respiration does is it provides energy for both growth and maintenance for different tissues in the plant, and also in leaves promotes efficient photosynthesis, meaning that when respiration is occurring and photosynthesis co-occur, they kind of feed back to each other in terms of substrate supply and then also energy. So again, this 60 gigaton value that I presented represents all plant respiration. And this can be divided into leaf respiration, which I focus on, and then also stem and root respiration. And leaf respiration is approximately 50% of whole plant respiration. So again, about 30 gigatons of carbon per year released. And if we think of this more at the ecosystem scale, the contribution of leaf respiration compared to, now I'm representing this as soil, is going to vary depending on ecosystem type. So if you think of a tropical rainforest that has multiple layers of leaves and a really high, what we would describe as like a high leaf area index, where there's many layers to the canopy, you're going to have a larger contribution of leaf respiration to the total CO2 efflux compared to soil. However, in a system like the tundra, where you have a very low leaf area, so fewer leaves per unit ground area, compared to the soil, of ecosystem respiration, you'll have a bigger flux coming from the soil than leaves. So the contribution of leaf respiration is going to vary with ecosystem type. And what my research focuses on are these like major sources of uncertainty in how respiration is modeled and represented at regional and global scales in different models. And my goal overall is to present the short and long-term temperature response of respiration, which is a major source of uncertainty, as well as the light inhibition of respiration. And while these are kind of might be thought of by non-respiration researchers as maybe minor phenomena in the metabolic physiology, the uncertainty when you scale up to the global scale could account for something as large as the tropical forest carbon sink. So again, if you're thinking of this 30 gigatons of leaf respiration a year, even slight differences of 5 to 10% are going to like accumulatively amount for a very large flux. So first, I'm going to describe the short and long-term temperature response of respiration. Sorry, I'm looking up a lot. It's easier than this. So generally, respiration increases at a rate, hits an optimum, or what I was describing with someone earlier, it's more like a maximum, because this isn't really a temperature you want to reach with plants. It's usually around 55 degrees C, and then decreases due to membrane denaturation. All right, so this is like a general response curve shape. And this short-term response is short-term on the order of minutes to days or so, so within that time bracket. So this is kind of like how it would respond to the difference between early morning to midday to night, and across a few days as well. And this response curve shape is based on enzyme capacity and also substrate supply. And a way to describe this response curve shape is with a concept called Q10. And Q10 is basically the temperature sensitivity, or the rate of response over a 10-degree interval. And you can think of it kind of, I drew these like little orange lines across to, that would represent like a 10-degree interval. But you can also think of it as like an instantaneous Q10 or a Q1, where you have kind of the derivative of this slope at a given point. So you know what is the rate of increase of respiration at that temperature. And historically, Q10 is modeled as equal to 2. And this is present in most Earth system models that are being applied to look at whole global terrestrial carbon efflux, and also at the ecosystem scale. But it's been known for quite a while that not only is this response not exponential, so it wouldn't be represented by a fixed value, but it's probably not 2 or consistently 2. So this is kind of like a known issue that hasn't been dealt with directly until now. So if we think about, that was a minutes to days response, a longer term response of respiration to temperature kind of falls under the category of acclimation. So again, if this is our short-term response of a leaf, so we're warming a leaf or cooling it, it's going to respond kind of in this manner, and then it'll hit it again like that maximum rate and decrease. And for leaves that are grown under colder conditions, or if you look at a latitudinal gradient and you're comparing tundra to boreal forest to tropical forest, and they have like colder growth conditions overall, or a treatment effect, for the same measurement temperature, you're going to find a higher rate of respiration in a colder grown plant, and this would be cold acclimation. And the opposite is true when you warm plants for longer term. So this, when I say long term, I'm thinking weeks to years. So this can actually happen on a pretty short time scale. So the difference between the trees outside in August and now are gonna, you'll see like an acclimation signal. It's that short of a term. And again, so under warm grown, so either experimentally warmed or plants found in warmer climates, when measured at the same temperature, you'll see this decrease in respiration rates at the common temperature. So the second source of uncertainty that I focus on in this metabolic process is the light inhibition of respiration. And this is also known as the Koch effect, and it's because it's attributed to Bezel Koch, who is the scientist that first found this phenomena in algal suspensions looking at rates of oxygen consumption. And this is from one of his earlier papers. So this has been known and published for over 60 years now. And light inhibits leaf respiration anywhere between 5 and 95%. So there's a really large variation in the degree of this inhibition. And again, thinking about this at a global scale, that translates into a really large flux of carbon from different, especially like heavily leaved systems like those rainforests. And the degree of this inhibition depends on environmental and biological factors. So what kind of growth form the plant is and whether it's warmed, et cetera, and higher nitrogen, things like that. And a neglect of this inhibition is going to lead to overestimation of ecosystem carbon uptake. So if we think of, again, knowing that leaf respiration is inhibited by light, if we think about like a daily carbon flux, so if you think of this as a single day, so midnight to midnight, and this is looking at net ecosystem exchange. So net ecosystem exchange is kind of like an instantaneous rate of carbon either moving in or out of a canopy system or any ecosystem, right? So a positive efflux means there's a net CO2 release from the system, and then again, this is kind of showing like nighttime and the yellow is daytime. So when light is out and photosynthesis is occurring, you have this net CO2 uptake, so it's a negative flux into the environment, and then the sun sets, it gets dark, and ecosystem respiration is the dominant flux from leaves and soil, right? So generally, what happens to model how much carbon is entering an environment, you take a value of ecosystem respiration measured in the dark because you can't parse them apart during the day because they're happening simultaneously. And you apply a temperature function, and you kind of add it on to net CO2 uptake. And you call this gross primary productivity, or all of the carbon that's coming into a system. However, knowing that leaf respiration is inhibited by light, we're actually overestimating GPP by anywhere from 15 to 20%. And this is especially true because the highest temperatures during the day, when you expect respiration to be higher, are experienced when the light is also out. So you have this inhibitory effect also occurring during times where you would model high respiration. So generally what I look at is I approach these two different uncertainties from two different scales, right? I look at this globally to get an overarching idea of common controls of respiration in order to better inform Earth system models and really predict and project how much carbon is entering and leaving environments. But I also look at these processes in more localized regional case studies to see within an environment how environmental drivers are changing these processes. So the first study I'll talk about today, or like one of the major questions rather, is how does this short-term temperature responsive respiration and the light inhibition of respiration vary across these diverse species and ecosystems? So this is kind of a much broader question that you need to look at a global level. So to answer a broad question, we need to survey as many plants as possible and to see what common controls exist and kind of reduce the uncertainty across species. So to answer these, I've conducted physiological surveys in different parts around the world using standardized measurement approaches. But again, that doesn't really answer what's happening on a ecosystem scale in terms of a changing environment. So the second major research question I have are what are the impacts of a changing environment on these processes and then in turn ecosystem carbon? So to answer that, for today I'm going to present kind of three major ecosystem drivers or environmental change drivers. An increase in nitrogen and phosphorus availability in the soils, overall elevated temperatures that we're seeing in many different sites, and then also phenology, which while not changing necessarily due to global change, there's lengthening of the seasons at both ends. So knowing how the seasonal variability of these processes affects carbon exchange is also kind of an unknown. So again, to answer these questions, you have to go to kind of specific places and look at global change experiments that are kind of pushing the system into new states. So specifically to look at questions around the temperature response, I conducted research at the Arctic LTER in the North Slope of Alaska and at a B4 Warms global change experiment in northern Minnesota. And then to look at light inhibition, I also was in the Arctic LTER in Alaska. And then to look at phenology, specifically I looked at the Harvard Forest LTER in central Massachusetts in a deciduous forest system. So what I'm first going to address is kind of more of this global picture. So how can we condense a lot of variation found in different species and when addressing this uncertainty of the short-term temperature response of respiration? So again, this is a globally based study using a large physiological survey. So this survey went to 18 sites. We used standardized methods of data collection that I'm going to talk about in the next slide. And overall collected data from 231 common canopy species from sites in South and North America and Australia and one site in Europe. And we collected replicate curves for each of these, so there was 650 replicate curves overall. Excuse me. And to classify this broad variety of species that were part of the survey, they were kind of partitioned into different categories. And one is representing plant functional types, which you can think of as these broad categories of plants. And plant functional types are often used in Earth system models to kind of maybe be a little more specific about a region's vegetation. And we also categorized all of the species to the biome where they were collected. So there's 7 different biomes and then 7 plant functional types included in the analysis. And I wanted to acknowledge the graduate students that I mentored during this data collection. So I didn't go to all of these sites. I focused on the northern latitude sites and worked with Sophia in Umeå, Sweden, Nur in northern, who's a graduate student at Australian National University in northern Minnesota, and then Angie Patterson at Black Rock Forest in New York, in the Hudson Valley. So specifically, the research approach, which I'm not going to go into too much depth about the measurements we made, but we took leaves from all of these different species, dark adapted them, and then increased the temperature using this Peltier-controlled system, which was plumbed into an infrared gas analyzer, the Lycor 6400, which some of you may be familiar with, and then warmed these at a degree a minute from about 10 degrees to 65 degrees or so. And this gives us kind of unprecedented high-resolution response curves. So for instance, if you were looking at older data on a similar question, you would probably have 5 points between 10 and 45 degrees. And now we're getting 2 points every degree, which is really allows us to do a much more fine-scale modeling work. And for this, because the data being used to this is used for kind of broad-scale applications of Earth system models, I focused primarily on this 10 to 45 degree window of response of temperatures, because that's where the temperatures that most plants experience through the year globally. So having all of these 650 odd curves, 673, the first question I had to address was what model best fits the data. So there are a lot of competing temperature response models of respiration. And again, this exponential fixed Q10 is one of the most prevalent in these larger carbon models. So all of these data were fixed or were fitted to these competing models, and we found the second order polynomial, or a log quadratic, which is represented in the green, had the data fit the model the best, or the model fit the data the best across this entire temperature range. And then these other models, the Lloyd and Taylor, which is often used in soil respiration, and then the Arrhenius, and this is a universal temperature dependence and exponential fit, fit relatively well in the higher temperatures, but really diverged at these lower temperatures. So this is kind of like the functional form of the model that we used, where it was natural log transform respiration is equal to a temperature function, a polynomial temperature function. And again, this was applied to the whole data set, and then species means were calculated, and then within those 231 species that were categorized by the plant functional type and the biome, we looked at how the data, or assessed how the data responded across temperatures. And first, what I'm going to show you is kind of a plot looking at the biomes. So this is, on the y-axis, we have the respiration, so natural log transform respiration, and then on the x-axis, the leaf temperature, all right? So this A panel here is actually measured data, and these are, so TU is tundra, boreal forest, et cetera. And tundra being the coldest, you can see actually has the highest rates of respiration, which kind of agrees with that idea of acclimation, right? And then the tropical rainforest lowland, which is French Guiana and the Peruvian Amazon, which were the hottest climates, also have the lowest. So you can see this even when measuring very different plant forms. So what we're looking at here is kind of these species means. So everything that was a tundra species is now averaged into this response. And then on the B panel, this is the averaged A, B, and C coefficients from all of those plants included in the tundra and projected, all right, as a model here. And if you look here, the intercepts definitely change, and those change due to like the long-term climate variability and also plant growth form. But what really stands out, if you've been looking at these response curves forever, is that the shape of the response curve is conserved. So you're seeing this kind of increase is happening in very different biomes at a very similar rate. And this is also true when just looking at the modeled projections of the data. So there's variation in offset, but a consistent curve shape. And when you look at plant functional types, this also stands out. So here's C3 herbs, which are generally known to be have a very high energy demand for growth. And you can see they have the highest respiration values as well. And again, there's this variation in offset, but the shape of the response curve is roughly equivalent and statistically equivalent. And similar with the modeled data. So overall, the short-term response of respiration to temperature is common across PFTs and biomes. So statistically, those curve shape parameters are similar across these broad categories of different plant species. So if you look at the global mean data, so this is all the species data averaged into a single response in the light blue. It overlays almost perfectly on the global polynomial model, which is the average of these two response curve terms, the B and C term. So you can see the model and data represent each other very well when we're talking about 231 species from 18 different sites. And if we think about Q10, or this temperature sensitivity of respiration, this declines. So again, if a Q10 was 2, an exponential response would be a fixed value across the whole temperature range. We're actually finding a decreased sensitivity of respiration as temperatures warm. So this is, next to the yellow star is the model using these, this is the average B and C terms. And knowing this shared response, you can kind of plug in these values to calculate respiration, leaf respiration at any given temperature. And you can do this either by using our species database that was published with the paper, or just from having a single known reference value and reference temperature. So again, the driving question of this study was to think about how we can condense global variability in different climate and growth form variation and see how that would affect models of carbon in different systems. So this is kind of a projection of when you change this Q10 of 2 paradigm that's been used in a lot of Earth system models to this new global polynomial model, where's most of the change occurring? And it's occurring in the cold latitudes or in montane regions as well. All right, but this doesn't tell us about how things are happening on the ground in terms of a changing environment. So first I'm going to talk about the temperature response and its impact or its effect on nitrogen and phosphorus, increased availability in the Arctic, and as well as long-term warming. So this is especially important when we talk about Arctic ecosystems because under warming we're getting an increase in the active layer in the soil, both in terms of volume and also length of time when things are warmer. And this increased active layer is going to have higher microbial activity and nitrogen and phosphorus mineralization, allowing plants to have a bigger available pool to uptake this nitrogen. And this is closely associated with the woody shrub expansion in the Arctic. So for this study, I looked at these long-term passive warming and fertilization treatment plots that have been treated for over 20 years in tussock tundra, and specifically looked at Betulinana, or dwarf birch, and Eriophyrum vaginatum, or cotton grass, which is a woody shrub, or Betula is a woody shrub species. It's ectomycorrhizal and it's deciduous, whereas Eriophyrum is a tussock sedge. It's non-mycorrhizal and evergreen. And this is collected in the same manner as the global study, where you have a high-resolution temperature response curve between 5 and 35 degrees, looking at respiration in the dark on a mass basis. And if we look at Betula, which is a control, under control, or a WG, which is long-term warm-grown, and then Eriophyrum under those same conditions, you see no acclimation. So acclimation would be an adjustment in that offset of the rates. And you can see for Betula, the circles, there's actually a complete overlay. So there's no acclimation. But when you look at Eriophyrum, at the same measurement temperature, you have a reduction in rates under long-term warming. So there is an acclimation effect in Eriophyrum. And what this means is there might be an ability of Betula to maintain a higher metabolism, so maintain these high rates even under long-term warming effects. And this might give it an advantage in terms of growth because it has more energy resources across this whole temperature range. So thinking about just Betula, so this study focused on just Betula in control, greenhouse, so again long-term warming, nitrogen and phosphorus, and then a compounded effect of nitrogen and phosphorus and warming. If you look at, it's kind of hard to because they're so close, but the control and greenhouse are kind of in here. And again, we see no acclimation effect. So it confirms the previous study. And we're also seeing this no adjustment under long-term warming. But when you add a ton of nitrogen and phosphorus to the system to kind of mimic that global change response, you see higher rates due to this increased respiratory rate at any measurement temperature under fertilized soils. And if you compare just the nitrogen and phosphorus to this nitrogen and phosphorus with long-term warming, you see an adjustment. So there's an actually acclimation effect between this black square and then the white diamond, I guess. So there's a nutrient-dependent acclimation, but not just under business as usual with warming. So if we were looking specifically just at warming effects on a different kind of system, so the Before Warmed study system looks at seedlings under warming and drought in northern Minnesota, specifically at the boreal temperate ecotone. So this ecotone is kind of where those two ecosystems are meeting. And then the global change experiment sets up kind of like a randomized block design of control warming of elevated 3.4 degrees below and above ground, and also drought and a compounded effect. Here's kind of a picture of this here. So they're infrared warming lamps and then buried cables to get the below ground warming. And then for the drought effect, there are these kind of rain out shelters that you can kind of like roll back during certain rain events to remove a proportion of the annual rainfall. So for this study, which again was based on seedlings, not adult trees, I looked at two more northerly species. So this is kind of a latitudinal gradient, where this middle stripe is kind of where the experiment takes place. All right, so it's a common garden experiment for these, with two northern species I focused on, white spruce and paper birch, and then two more southern species, white pine and red maple. And these are also paired in terms of their two needle-leaf conifers and two deciduous trees. So looking again at this temperature response of respiration for the needle-leaf, broadleaf, and southern range and northern range species, you can see if this is what acclimation would look like, an adjustment to rates downward with long-term warming, you see this only in the white pine and white spruce, right? So there's an adjustment with warming, and then slightly with paper birch to a lesser degree. And we don't see this acclimation effect in Acer rubrum, which is the broadleaf, most southern range species. And what this tells us is that there is acclimation in northern and needle-leaf species, but not the southern species. And this might be an adaptive response of climate stress. So if you're at this southern range, so this is at the very southern range of the northern range species, but under warming, it might be pushed into kind of a stressful environment or a stress response where you need to readjust your metabolism. And when we think about this on an ecosystem scale and how it's affecting growth and community structure, we see that these same white pine and white spruce are reducing growth. So the light blue is control with drought, warmed, and then warmed with drought is the dark red. And so these two needle-leaf species are actually reducing their growth when you measure stem volume as a proxy for growth. Whereas the broadleaf species, even the one that had some slight acclimation, are not really reducing their growth. So there's definitely kind of this growth form effect as well. So when thinking about the short and long-term response of respiration, we see a common short-term response globally when looking across different plant functional types and climates and treatments. But there's a metabolic downshift, so this acclimation that we're observing at plants at thresholds, specifically Eriophrum in the tundra that might be kind of a victim of the woody shrub expansion, and the northern range Minnesotan species. And this might be pushing for the woody shrub expansion again and a southern range expansion into the historically boreal area. All right, so next I'm going to focus on this other major source of uncertainty, the process of, or the phenomena of light inhibition. And I'm going to look at these same kind of long-term treatment plots of fertilization and long-term warming in Arctic Alaska. So the reason that this is so important in the Arctic is because, as you can see, this is taken after midnight. The sun doesn't set during the growing season. So this light inhibition can occur down to 5 micromoles of PAR, which is very low light for a plant. So you have inhibition basically occurring for the entire growing season, the snow-free growing season, where there's no real darkness. So the respiration in the light is really the only ecologically meaningful measure of respiration in vegetation in the Arctic, period. And again, neglecting this is going to lead to inaccurate estimations of total ecosystem carbon exchange. So this study kind of approaches 2 questions. Does leaf level metabolism affect ecosystem change in a future tundra? And does altered carbon exchange that we're seeing in this light respiration and photosynthesis promote shrub expansion? So exactly, is there like a metabolic advantage to woody shrubs over the Eriophrum species? So I'm not presenting photosynthetic data here, but generally, and this can be observed, Betula has statistically higher rates of photosynthesis almost under almost every condition compared to Eriophrum. And it has a greater inhibition of respiration in Betula under warming. So Betula compared for a control compared to warming, the inhibition is greater under warming. So it has kind of more carbon to work with during that time. So overall, what we're seeing is this enhanced carbon gain efficiency. So carbon gain efficiency is a metric that I used for this paper, where it's basically you can think of it as like how much carbon is staying in the plant versus being cycled instantaneously. And under control, Betula areophrum, and this is rubus, the third species I'm not talking about, have equivalent carbon gain efficiency, but under warming, Betula has a significantly more carbon to work with under this kind of like altered environment. So again, it's not acclimating, but in terms of the actual carbon fluxes, it might have more. And this is also interesting under increased nitrogen and phosphorus. So Betula is in the black circles and areophrum is the open circles. And this is leaf nitrogen and leaf phosphorus content or concentration on the x-axis. And this is respiration in light, again, that like true value of respiration in the Arctic. And you can see areophrum isn't really sensitive to increases in fertilized soils in terms of taking that nitrogen, allocating it to the leaves, and using it for metabolism. Whereas compared Betula, both under nitrogen and phosphorus additions, these are was under a 3-part graded addition study, you can see it's very sensitive. So it's not only responding by taking up that nitrogen and phosphorus, it's allocating it to the leaves that can then be used for carbon metabolism and energy. So there's definitely a competitive nutrient uptake that we're seeing in the shrubs that we're not seeing in the areophrum species. And this is visible just walking around these treatment plots where you see, and I don't know how clear this can be, but this is like, these are like very big shrubs for the Arctic, and they're loaded up with fertilizer, and then this is in the passive warming tent here that raises degrees, maybe 3 to 4 degrees over the summer. So these huge shrubs. So there's definitely like this carbon exchange advantage, and it's likely shifting ecosystem and community structure. And we can measure this and put values to it. So under a control plot, areophrum and Betula have equivalently the same, making up about 20% each of the community composition in these plots. But when you warm the system, Betula increases by about 50%. And when you add nitrogen and phosphorus to the system, it increases about 200% compared to the control. And it increases as areophrum is decreasing. So it's kind of at the detriment of the woody shrub, mostly because it's being shaded out and it has a much better nutrient acquisition abilities. So overall, there's this metabolic efficiency in the woody shrub Betula over areophrum, and this might favor woody shrub expansion in the Arctic tundra. And this ability is, again, kind of pushing into shrub invasion and expansion in the Arctic tundra, which has a lot of downstream ecosystem environmental effects, including earlier snowmelt, decreased albedo due to earlier snowmelt, and kind of like a different surface roughness of the shrubs, winter soil warming, and increased permafrost thaw. And collectively, these have the potential to destabilize this gigantic soil carbon pool that exists under the vegetation and can feed back to the general climate system. So the final case study that I'm going to talk about is more focusing on phenological effects, or kind of like a seasonality in this light inhibition in a deciduous system of hardwood forest. So I'm looking at these seasonal effects of daytime respiration again, so not just the nighttime value. And a recent study, this is kind of being brought to the forefront in terms of ecosystem work, because a recent study used a novel technique, JWxrY, who's also working at hardwood forest, using isotopic flux partitioning. So using an eddy covariance tower that measures that net exchange, but real-time isotopic fractionation of the flux values coming in and out of the system. And from this, they found that daytime respiration was reduced. And if you were study leaf respiration, you'd be like, yeah, we know. But we're not using eddy covariance to study this. So a downside of that study is that it's difficult to parse apart species effects, environmental effects, and seasonality. So you still need to kind of look at things at the leaf level to be able to say more specific predictions about the ecosystem level. So in this study, I focused on Quercus rubra, which is the dominant canopy species in the tower footprint, and looked at respiration in the light and respiration in the dark over the growing season, and 2 different canopy heights. So this is just kind of giving you an overview of that effect. So if our light this is kind of a ratio of R light to R dark. So if R light to R dark is 1, there's no inhibition. And if it's 0, that would be full inhibition, right? So what we're seeing is that there's definitely an inhibition effect. And it's roughly across the season about 25%. So enough to definitely be mindful of when we're thinking about huge carbon fluxes. And the least inhibition that we see is during the early season when there's a really large growth demand for energy. So if you're leafing out every year, you need a lot of energy, and a lot of that energy is coming from respiration. And then also at the end of the season, a little less inhibition that might be associated with senescence, where again, you need energy to allocate some of the resources that you're going to use again for the next growing season. So while photosynthesis is kind of deteriorating a little bit, leaves are changing, mitochondrial is still providing a lot of energy for that movement. And if we think about this over the course of the growing season, this is just kind of a simulation I did where this is from May 1st to September 30th, so roughly the hardwood forest growing season. And then taking into account, this is instantaneous rates of respiration on a 15-minute interval, that takes into account a short-term temperature response, short-term or 10-day acclimation effect. And then the black values are just looking at respiration in the dark. And then the orange values on top are looking at respiration in the dark. And then any time that the sun was out or light was greater than 0, basically, I think I set the cutoff at 5 micromoles, there was an inhibition effect on respiration. And collectively over the season, this led to a 20% reduction in cumulative leaf respiration leaving the system. So again, when we're thinking about whole forest systems, this is a lot to account for. And this kind of leads me to some next steps, because I think I mean, there are many, I've talked to some of you today about what my future goals are, and one of them would be to set up similar research at the Michigan Biological Station to look at R light and also kind of tie these into the eddy flux measurements at the multiple tower sites up there. And so this leads to scaling opportunities and also looking at different kind of successional gradients and how maybe a growth stage of a plant is going to affect its energy demand, et cetera. And this would also have an opportunity to tie in different measures of gross primary productivity, which can be measured above the canopy with solar-induced fluorescence, and also kind of ground truth with leaf-level light, fluorescence in the light. And then following, so I presented to you this global study I worked on with the temperature response, and I'm also currently working on, but I'm not going to go into depth on today, a global R light database. And with the same goal of kind of reducing a lot of this variation that we're seeing across species and environment and seeing if it can be not simplified, but kind of modeled in a way that would be helpful for larger models in terms of accounting for this inhibition effect. And currently I have over 1,100 individual response curves from a lot of the same sites from the first survey work I showed you. And then finally, some other future approaches that I'm thinking of have to do with, again, these global change and environmental change questions. And a lot of the research I did on global change experiments focused on long-term constant rates of warming. So we're increasing temperatures by 3 degrees or 5 degrees or something like that. But we're consistently increasing across the season. But when we talk about these highest months, record month temperatures, we're actually thinking about collective heat events that are happening around the world. So a heat wave that increases by 5 degrees C for a week in June is going to have a different effect than this kind of sustained elevated temperature through the season. So you're going to get these short-term acclimation effects and also kind of overlapping with a drought signal. So I'd like to kind of tease those apart, both doing field work and then also kind of much more controlled in growth chambers. And then finally, a lot of the work I did and that I presented today were in kind of these intact, quote, pristine environments of forest and tundra systems, which might not be where a lot of the change is being experienced when you think about human development. And especially in terms of higher CO2 in nitrogen deposition. This is happening also in forests, but maybe much more rapidly in like the kind of like urban and suburban and rural mosaic. And I see a lot of potential tapping into the Ameriflex network, just even in Michigan and northwestern Ohio as well, as kind of like a scaling opportunity to look at more kind of like land use change effects. So finally, I kind of hope that I brought attention to and maybe educated everyone on this short and long-term temperature response of respiration and the degree of uncertainty that it introduces to models, as well as the light inhibition of respiration and how it can change across environments, both when we consider it at the leaf level scale with which it's measured, but also how it can be altered under different environments at the ecosystem scale and maybe promote things like woody shrub expansion in the Arctic, or different range shifts in boreal and temperate forests, and then how we can kind of condense that to look at it on a global scale. So in conclusion, globally, again, the temperature response of respiration is conserved and common across biomes and plant functional types, which is a finding that will hopefully improve global projections of plant carbon in current and future environments in Earth system models. In the Arctic tundra, where I looked at both the temperature response of respiration and the light inhibition, this might allow for a metabolic advantage in terms of like energy and carbon to promote shrub expansion under future conditions like these high nitrogen and warmed environments. At the boreal temperate ecotone in Minnesota, we're again seeing this metabolic adjustment of acclimation in northern ranged and needle-leaf species, so kind of a selective acclimation effect in these more potential threshold species. And this is going to lead to growth limitation under warming and seedlings, so that might have like a long-term demographic effect of what those forests will look like. And then finally, in temperate forests, I looked at inhibition of respiration through the season and how it lowers this overall ecosystem carbon estimates when we think about using respiration to calculate gross primary productivity. Yeah, and then I have a lot of people to thank because this is, you know, a few years of my life doing all this. So a lot of people at my home institutions and grad students and undergraduates that also helped with the research. All right, that's all. Yes.
I know some folks have to.
It's 5.
But for those who stay, I'll let Mary field her own question.
Yes.
Yeah, so this question comes out of very deep ignorance. Hear that and help educate me a little bit. So I was very interested, especially in the beginning part of the talk, in the work we've done to look at the leaf responses to temperature, and then the regularity you see in those things sort of embolden you to kind of scale them up, extrapolate up to larger scale.
Mm-hmm.
But it occurs to me that that's a really difficult thing to do in general, and that there might very well be different extrapolations in different biomes, different kinds of scriptures. So I wonder, is it possible to ground truth those extrapolations to some degree? If so, has it been done?
So when you're saying extrapolations, you're talking about the
scaling it up to
right. Yeah. So the scaling that's applying. So when we're inserting like this global model, right, that's found to be common across all these different ecosystem types based on the species that we measured at that site, and applying that into a model is as simple as, I mean, it's not simple, but it's changing the temperature function. So I think what you're asking is like if we took a ecosystem measure of this response, or if you're,
So it's easy enough to stick it into the model.
Right, yeah.
But then the question is, does that actually predict what's going on at the higher scale? And is it possible to check?
It's difficult to check just because of the way that ecosystem carbon, like net ecosystem exchange, is measured. So it's measured as kind of like this bulk flux and not necessarily partitioned into leaves and stems and soils, et cetera. So you just see like how much carbon is leaving at night. And that will have its own temperature response that's going to have soil and stem and, you know, like woody tissue and heterotrophs and all this stuff kind of factoring in. So it would be difficult, but it's, you would just have to partition those separately, I guess, to ground truth. So you could take a canopy response, but also kind of the same measurements we already took. So it'd be, yeah, hard to tease those apart. People are doing work looking at whole temperature responsive ecosystems by kind of taking the eddy flux data and then modeling that across temperature. But that doesn't parse apart the different pieces. That's can be done by, like, you know, cutting trees so you can kind of do autotrophic and heterotrophic soil and responses, but you'd have to kind of take each component individually. Yeah. Yeah.
So again, sort of looking for generality of costing, you had taken arguments that you had very different responses between the shrub and the sedge, but is that shrub representing small shrubs or the sedge with other graminoids?
Right.
Are those, and if so, why? What is it about the growth ones that might give you the different
Yeah. That's, so to answer the first part of your question, I'm using them as kind of proxies for their, like, plant functional type, or like this general, like, shrub class and graminoid class, right? And at least in that part of Alaska where I was measuring, those two species account for like 40% of the ground cover. So whether or not they represent other shrubs, they represent, you know, 20% of vegetation. So it's like a pretty, both of those, and not just in Alaska, but they're found like circumpolar, like Sweden, et cetera. And so they're important as kind of like these iconic, like, poster species of the Arctic. And then the second part of your question was how we can, like, use those to, or like how representative they are. Or?
No, it's both how representative are they, and it's because what is it about being a shrub?
Oh, right.
Or is
Yeah.
it that particular shrub that would give those differences one to the other?
Yeah, so I thought about this a lot when looking at the data, and the betula, for instance, like I mentioned, is ectomycorrhizal, so it has this kind of advantage in terms of taking up nutrients from the soil that the graminoid does not, right? So it kind of already has this nutrient acquisition advantage, and then also it's deciduous. So deciduous species often have higher rates of respiration than evergreen, so the graminoid is an evergreen. And just faster growth rates, the need, especially in the Arctic where the growing season is like 2 months long, 2.5 months long, there's a fast pulse of leaf out where you need to expand leaves, assimilate carbon through photosynthesis, but also have energy to do that that's coming from respiration. So you're going to have, and this could be the same looking at, again, the Minnesota case study where you're looking at needle-leaf trees and broadleaf trees. You're going to have general, like, higher basal rates of respiration just due to that kind of like growth demand of putting out leaves really quickly in the early spring or so. So yeah, I do agree that there's probably something about their growth forms that are like driving those differences. Okay. Mm-hmm. I think I dug a little more deeply into like algal responses when looking at light respiration. And like the first studies, like this Koch effect is based on algal suspensions. So like the light inhibition is definitely seen in marine algae and I think like freshwater aquatic algae as well. So that response is there. And in terms of acclimation, I would believe so. I haven't read anything recently, but a lot of systems subjected to long-term warming, like even like soil has an acclimation response under certain systems where there's just kind of a downshift to either be more conservative about energy production or carbon loss. So yeah, I would expect ocean systems to have similar, maybe to a different degree, because they're facing kind of But I mean, like the warming pressure, I would assume, I don't assume it to just keep getting, you know, be a linear direction with warming, but rather some kind of like adjustment over long terms. I don't know like if that's going to happen on a 10-day basis or like a 10-year basis, but I would expect it to happen.
I'm really interested in thinking about what's going to happen in the boreal regions as they're, experiencing warming circumstances. And I tend to think about what happened back in Eocene when there were actually evidence of tropical forests, tropical types of forests that extended to those far northern latitudes. But I wonder, is there going to be a limit to the amount of deciduousness geoscience you can exist in boreal areas based on the very minimal amount of daylight during winter periods? I mean, is that going to be a limiting factor? Like there's months where there's hardly any daylight.
Right.
Would trees still need to respire? Is that going to, the lack of light going to be a limiting factor to the expansion of deciduousness in this forest?
I'm not sure how limiting it would be. Again, kind of referring to the Arctic data, I mean, there are deciduous plants in the Arctic that already have, you know, only 2.5 months of daylight, basically, and they're covered by snow the rest of the time. So the timing might be different. Right. Yeah, it's dark but warm, yeah. Yeah, we were talking about this morning with Catherine, and like one of the kind of a early morning brainstorming session thought of like, I would assume deciduousness would still, I mean, like there's a energy conservation benefit of having needle leaf or evergreen leaves because you're not investing every single year in all of this carbon and energy in the same product. So there could be a retention, but I don't know if, I don't think deciduous trees would become less deciduous, I guess, under this warming and darkness. Yeah, and which happens with needle leaf trees anyway. You know, like it's very cold, so the rates are very low. Like in, you know, sub-freezing, and you see a spruce tree outside, it's still respiring, but it might be like, you know, barely measurable because it's so cold. If it was warmer, it would, you know, probably be a lot more detectable, but they're still respiring all year.